All right, well, welcome to our March meeting. And we're very excited to welcome Dr. Frank Rinkevich this evening. He's going to talk about Amitraz resistance and Varroa mites. We also have a presentation by our very own Brian Fackler about yellow jacket management. The B School is up and running. Susie has a lot of active members this season. And as you may recall, our B School is designed for beginning beekeepers. Susie focuses on detailed and actionable information. There are 4 two-hour lessons for a total of 8 hours via Zoom, live or recorded. And new this year is a 2-hour apiary session with Susie and Jana out at the apiary at Green Acres. It's $110 for the 4 classes and the apiary session, and it includes the 3rd edition book by Dewey Karen called Honeybee Biology and Beekeeping. The experience of the beekeeping school is supercharged when you activate and engage with the Bee Buddy map. The next Bee School date is on the 9th from 7 to 9 p.m. and it continues every Thursday through May. The class covers intro to beekeeping, equipment, obtaining and installing bees, honeybee biology and behavior, inspections, Varroa mite biology, integrated pest management strategies, seasonal management for the Pacific Northwest, and of course, the honey harvest. Next month, we'll have Dr. Ramesh Sagili. He'll talk about Varroa and nutrition management. And if you have any suggestions for new topics, you can send an email to me or anyone on the board, and we would love to consider that. Without further ado, here is Dr. Rinkevich.
All right. I'm going to start sharing my screen. All right, so is my slideshow up there?
It is.
Okay, great. It does work. Well, thank you very much for inviting me to speak to y'all. This is a good opportunity for me to get out and teach people about what I think is a very important issue in beekeeping, especially on the commercial level. So whenever I give this talk, I always try to remind people that you don't have Varroa because you're a bad beekeeper. You have Varroa mites because you have honeybees, right? So it's one of those things that we need to be aware of the issue with Varroa and how that could be affecting your bees. And so the reason what I want to start off with is that you have to consider bees as pets or livestock that require veterinary intervention, right? So if your dog has fleas, you don't just say, oh, they'll figure it out. They'll get rid of their fleas on their own. No, you treat them with a flea collar. Same thing with cattle. If you raise cattle and they get ticks or things like that, you could use a bath, insecticide bath, or the treated ear tags. You have to do some kind of intervention to take care of the problem. And honeybees are no different. They are considered livestock by the USDA, and so they should be treated just like any other pet or livestock. And so the reason why Varroa are really important is because they are the major honeybee parasite. Just that name, Varroa destructor, that doesn't sound cute or nice or friendly. It sounds really bad. And you can see here the ugly Varroa. Well, actually, it's not too ugly. It's actually kind of nice looking, but on a honeybee here. And this is my opinion based on what I think the literature says and my own research. About 70% of the honeybee losses across the country are due to this mite and its associated viruses. And I'm starting to feel more and more confident in that statement every day that we have more research. So I say that, and that's what the science says, but also beekeepers know that Varroa is a really big problem. So this is the annual survey that's performed by the Bee Informed Partnership. And on the bottom here, they have the different reasons for colony losses. So we have Varroa, queen issues, starvation, no losses, weather, and a number of other things. And then it's divided into 3 different categories: backyard beekeepers who have less than 10 colonies, sideliner beekeepers who have between 10 and 50 colonies, and the red bars here are the commercial beekeepers who make a substantial portion of their total income off of bees and typically are considered over 50 colonies. And as you can see here, especially where I'm concerned is, especially with the commercial beekeepers, that most of them are saying Varroa is a big issue that's causing their colonies to become lost. So this right here means that the beekeepers understand that Varroa are a problem. Well, why are Varroa a problem? I mentioned that they are these parasitic mites. Well, they affect honeybees in two different ways. The first thing they do is they feed destructively on fat body. And fat body in insects is analogous to the human liver. It makes proteins and hormones and peptides and neurotransmitters. And it's basically the Swiss Army knife of insect organs. And where it's at is just under the abdomen right here. And these blue and red dots show the location of feeding sites of Varroa on the adult honeybee. So you can see they're feeding on the abdomen, right where that fat body is shown. And there's a bunch of other observations that support that, yes, Varroa are feeding on this organ, this fat body. Fat body is really important for overwintering survivorship. That's why colonies heavily infested with Varroa typically die over the winter. Okay, so it's really bad. It feeds on this really important organ, but it also acts like a little syringe for different viruses. So Varroa transmits two of the most devastating honeybee viruses, such as deformed wing virus, as you can see over here on the left. Typically, when it gets to very high levels in the bee, it'll express these crumpled wings, as you can see here. A lot of deformed wing virus infection is asymptomatic. So most of all your I could say this with pretty good confidence, that all your bees have deformed wing virus. It's just that Varroa cranks up the levels of deformed wing virus. So that's one thing. And then also too, chronic bee paralysis virus. Bees typically with this virus will be the shiny, oily, hairless sort of look to them. And they crawl around real slow, like the name paralysis virus implies. So these two viruses are directly related to Varroa levels in your colony. More Varroa, more viruses of deformed wing and chronic bee. Well, so beekeepers know it's a problem. We just talked about how they affect honeybees. So a lot of times beekeepers will use miticides to manage Varroa. And they can be any of these products up here that contain the active ingredients Amitraz, and that's what we're gonna talk about more tonight, oxalic acid, formic acid, and thymol. And so they work, they're mostly used because they work. There's no question about that. When used according to label, remember the label is the law and it's also instructions for high efficacious applications. They work until they don't, right? And so when I started this project, it was really aligned with my background. My background is in insecticide, the origin and evolution of insecticide resistance. So now just taking that paradigm and shifting it on its head for Varroa mites. And so I've been working on this project for about the last 4 years. And so I'm just going to tell you about what we've been finding. So we're going to start off by telling you about the project history and the scale. Where have we gone and what have we done? What is the prevalence of Amitraz resistance? Is it expanding? Is it going away? Staying the same? We'll find out. And then how we could use the results of the resistance test to come up with a treatment decision matrix. So what you should do based on the results of this test. We'll talk about the geography of resistance. Can you get resistant Varroa from your neighbors? Always an important question. And then some of the future directions that we're going to be working on. And through tonight's talk, if you have any questions, just interject. I want this to be more of a conversation, more the facilitated conversation, as opposed to just me sitting here talking.
And
all right,
Frank, I'll
yeah.
monitor the chat, and if I got any question, I'll chime in if I need to.
Outstanding, great.
Cool.
So the way that we determine resistance is by using the APIVAR resistance test. And this test runs by, we just take this 1 quart soup container that you get from a deli, right? I literally get these from a restaurant supply place. And we glue a square of APIVAR. You see that square right there? That's APIVAR. It's a strip cut as long as it is wide. And then we glue it to that container. We then put about 300 bees from brood frames into that container and seal it with a screened lid. On the other side, this is a screen. We then hold this over this tray with petroleum jelly on it, and then count the number of Varroa that fall off within 3 hours. 3 hours is the time it takes for 100% of the susceptible Varroa to drop in this test. So anything still on the bees after 3 hours is by definition resistant. And you can see what those Varroa on the tray look like. These dots here are the Varroa. So we count the number of Varroa that fall off in the tray. We then also wash the rest of the Varroa off the bees in that container to get the number of Varroa in the wash. We then take the number in the tray plus the number in the wash to get the total Varroa in the sample. And the way that we calculate the APIVAR resistance is we take the number of Varroa in the wash, the ones that are resistant, divided by all the Varroa that we find in the sample to get APIVAR resistance. So if it works like this, so let's say we have 5 Varroa in the wash, but there are 10 total Varroa. 5 divided by 10, 50%. 10 Varroa in the wash, 10 total Varroa, 100% resistance. This is super easy. It's not complicated. Now, some of my other friends say, well, actually, you have to do this and correct. I'm like, no, I know. But this is easy and it works just as well. So it's a very simple method. And so where have we done this testing? We've sampled lots of colonies. And so all these dots here show the location of apiaries that we've sampled in the last 4 years, mostly focused in North and South Dakota, as you can see here, but really good representation in New York, Pennsylvania, and then again through the Carolinas, through Alabama. We don't have any from Oregon yet, so hopefully we get some of those Oregon samples on the list very soon. So, and also too, we've started sampling in Canada. So how many samples have we collected? And when do we collect these samples? We usually collect them in August through September because this is the longest time those Varroa haven't seen any miticides. And it's also the time when Varroa are typically at their highest population levels. It's the longest interval without treatment. And we also want to make sure they're not being actively treated with miticides, just that way we could avoid the confounding variables of miticide treatments. And that's exactly why we perform this test before any miticide is applied. And typically miticides won't be applied at this time of year, August or September, because that's when hunting season is. So as soon as hunting comes off, that's when they get treated. So that's why we're doing it at this time. And also there's a temperature sensitivity of test. It's kind of like Goldilocks. Can't be too warm, can't be too cold, it has to be just right. And the conditions where it's just right is about August through September. So, all right, so how many places have we sampled? So in 2019, when we first started this project, it started off kind of small. We started with 7 beekeepers, and we sampled 11 apiaries in those operations for a total of 83 total samples. So we started small. And so this is the data set that we first published. Well, we started talking about this. Now we actually documented that resistance was a thing. And then COVID hit. And so we had to get a little creative in our sampling in 2020. And that's when we worked with the Apiary Inspectors of America, and I sent them the kits that we use for testing. And we were able to do some sampling in 2020 despite not being able to leave Louisiana. So we did increase our number of beekeepers, apiaries, and colonies, about doubled with their help. So I still work with them to this day. Then in 2021, we worked with the Honeybee Health Coalition and Jeff Williams at Auburn to, again, double our sampling effort. We're up to 27 beekeepers and 45 apiaries for 426 samples that year. And then finally, last year, I got hooked up with the Bee Informed Partnership. We spent 2 weeks together in North Dakota collecting samples. So we total in 2022 had 35 beekeepers, 72 apiaries, 700 colonies. So you can see here, every year we keep pushing that number up and up and up. And so hopefully this year we'll have over 1,000 colonies sampled across the country with good cooperation. All right, so what do we find? Is it all gloom and doom? Is this the end of beekeeping as we know it? Well, I don't think so. I think there's actually some good things that we found in here. So in total, we sampled 1,380 total colonies in the last 4 years. And out of those 1,380, we found that 291 or 21% had 0 Varroa in them. That's actually really good news. That means that there are a lot of people, 21% of all the samples that we collected, we couldn't even determine resistance because you didn't have any Varroa. That's a good thing. So some people say, oh, I'm sorry I didn't get any Varroa for my bees. I said, no, this is actually really, really good. Tell me what you're doing. So that's really good. The second thing that's also, I think, is pretty decent news is that we found out that 945 or 68% of samples had less than 10 total Varroa. So this is important for 2 reasons. One, if they have less than 10 total Varroa, I'm not super confident in assessing resistance or not resistant, because with small sample sizes, plus or minus 1 either way, it could really change your percentage in tenfold, right? It goes from 10% to 20% if you have 1 Varroa left on the bees. So with less than 10 total Varroa, that's actually kind of, it doesn't give me a lot of confidence in the assessment. The other thing is that is below damage levels in some stocks of bees. So this means that in 68% of the samples, we found Varroa levels that were pretty much under that damage threshold. All right, what do we find in terms of resistance? So we found bees where there were, Varroa mites, where there was 0 resistance, meaning that all the Varroa died during the resistance test. And 32% of samples had 0 resistance. So there's a lot of susceptible Varroa, Amitraz susceptible Varroa out there in the population, and we have to maintain those as a resource. But the bad news is we did find levels of resistance to Amitraz that would lead to treatment failure at the colony level. And that that resistance level threshold is about 30%. So if it's more than 30% resistant, that's going to be treatment failure at the colony level. And we did find that in about 29% of all our samples. So this is kind of the thing that we need to keep track on. Well, where is this 30% resistance threshold coming from? How do we get there? All right, so let me walk you through this figure here real quick. So on the bottom here, we have different apiary codes. And these are just anonymous identifiers that protect the identity of anybody who participates in this test. And I will never tell anybody whose Varroa I tested. So this is all anonymous. If you could figure this out, you should be probably working for the CIA. So what we have here is those apiaries on the bottom. And then on the y-axis here, we have apivar resistance. 0 is low resistance, 100 is high levels of resistance. And so you can see here, we have a bunch of colonies which we find no resistant Varroa. And then we have some intermediate scaling up. And then we get to this 30% threshold. And you can see all those ones above 30% are shown in red. Well, see these ones here with the hash marks? These are apiaries in which we sample the Varroa during an active Amitraz treatment that the beekeeper put on. And these are the resistance levels that we found in those Varroa. So these are the Varroa that are still alive during the active treatment. So that way, all those that were tested during the active treatment had more than 30% resistance. So that's how we come with that threshold for control failure. So that's the 30% resistance threshold for treatment failure at the colony level. All right, so when you run this test, you get two pieces of information. You get the apivar resistance levels, and you get your Varroa infestation. Hopefully, everybody is monitoring for Varroa on a regular basis, so you can get that information. But you can also get some qualitative information. Will apivar or Amitraz work in your colonies? So we have this broken down to 3 different categories of apivar resistance. Less than 10% resistance, I would consider to be susceptible. This 10 to 30% is intermediate, and above 30%, remember, that's what's going to be the level of resistance that leads to treatment failure at the colony level. So low, medium, high. And then for Varroa infestation, I express this in terms of Varroa per 100 bees or percent infestation. If it's less than 1, that's very low infestation. 1 to 5, I'm calling that medium. And more than 5 Varroa per 100 bees, it's a high level of Varroa infestation. And these categories are somewhat based off of what we know leads to treatment failure at the colony level, and also to what are damage thresholds for Varroa. All right, so let's look at the first section. So where you have less than 10% apivar resistance and less than 1 Varroa per 100 bees, is that we could confidently say that there's low Varroa pressure, right? Yeah, low Varroa infestation, and no Amitraz resistance. And then in this situation, I would recommend continuing monitoring and then treat as needed. You know, Varroa is low, resistance is very low. So when you treat, the treatment should be effective. Well, let's look at this situation where we have that low apivar resistance, but high Varroa infestation. So we have that situation where we have high Varroa, no Amitraz resistance. In this situation, Amitraz should provide complete control and be based on the resistance level, and the treatment should be imminent. So when you have this high pressure from Varroa, you probably want to get a treatment on ASAP. So that's what we'd recommend in this situation. What you don't want is this situation where you have high levels of resistance paired with high levels of Varroa infestation. Because in this situation, Amitraz will not provide complete control because of high resistance, and the recommended imminent treatment with alternative miticides, because Amitraz probably won't work. It's probably going to lead to treatment failure. So use something else immediately. And this whole thing is hatched out with different situations, and this is also available that I can provide as well. So that way, when you get the results of your test, you know what actions to take. So now you know your Varroa levels and you know your resistance levels. So the question I usually get from people is, can I get this from my neighbors? So this past summer, we spent a lot of time answering this question: is how does geography and apiary density influence distribution of Amitraz resistance? So the idea is that the closer and more densely these colonies are packed, the more uniform Amitraz resistance should be. So here we have North Dakota, if you can believe this or not. And this is the location of all the apiaries in the state based on the North Dakota Department of Ag. They have to register those colonies. And what we did is we looked at apiary density across the state on a relative level, where colonies, apiaries that are shown here in yellow represent high density apiaries in which there are many apiaries within a 5-mile radius. And then the apiaries in blue are low density, where there's very few apiaries within a 5-mile radius. So we use that as our density sampling gradient. But you can also see here we have these dots across the state, and these are occurring in different agricultural and geographic regions. So we have the whole state covered in terms of distance as well as density. And so that's what we have here. And the state is broken down into these different regions based off of geology and crops that are grown. So this is kind of how the state divides up its geography based on landscape use and underlying geography and geology. All right, so if we're asking this question, if density and geography are important to Amitraz resistance, some of the patterns that we should see is that the Amitraz resistance should increase and become more consistent at higher density. What does this look like graphically? So on this graph here, we have apiary density. 1 is the low density, where there's very few apiaries surrounding it, and 5 is where there's many apiaries surrounding the apiary that we're investigating. If this did matter, because that would mean that Varroa could travel between colonies easily due to foragers that are wandering between colonies, we would expect that at higher densities, they could move through easier. So what we should see based on this is that resistance would increase, you can see by the increased bars, but it should become more consistent. As you can notice here, the error bars decrease as we get higher and higher levels of resistance. It becomes more homogeneous. So this is what the data should look like if this was true. We'll come back to that a little bit later. The other thing is that it should be similar compared to the adjacent regions. So we should have regions where there's high levels of resistance next to areas where there's also high levels, low levels next to low levels. And it should be similar among beekeepers, because if this is homogenizing it based on density and proximity, then we should see a lot of similarity among beekeepers. All right, so let's look at the first thing I mentioned. As apiary density increases, we should see Amitraz resistance increasing and become more homogeneous. Well, that's what it looks like in theory, but in reality, it's not even remotely close to that. So again, these are our observations of Amitraz resistance. So as apiary density increased, we saw no relation to the levels of Amitraz resistance. Heck, at apiary density 3, we didn't even find any resistant Varroa. So you can see here, there is no consistent trend with apiary density. So it doesn't look like Varroa move between colonies easier at high concentrations, at high densities of apiaries. Now, it also looks like there's no influence of geographic distance. So, and the reason why that is because if you look here in the Northwest, it's the most susceptible region, but Varroa from the North Central are highly resistant. So some highly resistant Varroa directly adjacent to very susceptible Varroa. Similarly, down here in the southeast part of the state, we have high levels of resistant Varroa directly adjacent to very susceptible levels of Varroa. If geography mattered, these two values should be much more similar. So this is the preliminary analysis, and we'll have to still do more. But yeah, it doesn't look like you could get these Amitraz resistant Varroa from your neighbors. What can happen, and this is what I think is really driving the whole system, is that we see the highest amount of Amitraz resistance variation among beekeepers. So I think it's the way that beekeepers are managing Varroa and applying Amitraz that really lead to this phenomenon. I think it's frequent applications of Amitraz that leads to constant selection. So you can see here, we have beekeeper C, which has no Amitraz resistant Varroa, compared to beekeeper K that has extremely high levels of resistance. So the major factor that determines whether you have resistant Varroa or not is the beekeeper. And that's been the consistent theme that we've been seeing throughout the 4 years of this project. All right, so what have we learned so far, looking backwards? So we do know that Amitraz resistance is a fact, and it's the most consistent factor that explains control failure at the colony level. We've kind of did some myth busting when it came to people giving us explanations of why it doesn't work. Oh, it's old Apivar, it's gone bad. Nope, I've tested Apivar 7 years old and works just as good as stuff out of the package. Oh, something happened. I don't know. The only thing that can really explain it on a consistent basis is resistance. I've heard many things. So yeah, resistance is the most consistent factor, descriptive factor. At this point, Amitraz resistance is not a universal threat to the beekeeping world at this time. However, ... And I came to this realization is that over the last couple of years that we've been seeing this, we have been sampling more resistant populations more and more frequently. Now, I don't know if that is because we are working with people who are reporting problems more frequently, or is this increasing in frequency to the level that's reflecting in the data? I can't say either way. However, I do hear anecdotal reports from beekeepers suggesting that that is the case where it is becoming more prevalent. So we need to start thinking about beekeeping in a post-Amitraz world before it's too late. Apivar resistance can predict Amitraz treatment success and prevent costs associated with Amitraz treatment failure. So in this, when you run this test and the Varroa are susceptible to Amitraz, you can have very high levels of confidence that Amitraz is going to provide complete control. And I see that time and time again. I have a lot of data to show that shows that's the case. I don't have as a lot of data when they turn up resistant in the test, because a lot of times beekeepers aren't going to want to apply something that they don't think will work. So I know when they're susceptible, Amitraz will provide control. Amitraz resistant, I don't have a lot of data to back that up, but I do know it is when they are susceptible in the test, they're susceptible at the colony level. So that is good. I didn't talk about this very much, but one of the things you can do to help mitigate Amitraz resistance development is by rotating miticides. So using things like oxalic acid, diamol, or formic, it could help overcome Amitraz resistance and impede the development of resistance. And I'm working on a big paper talking about that. All right, so where are we going next? Where are we going next couple of years? Varroa genomics. This is the big thing that I'm working on. We can finally get Varroa out of it. DNA out of Varroa. So we're doing genomes of nearly 500 Varroa to develop genotypic tests. So we can test for DNA for resistance. I think we are pretty much close to publishing that. And I think the future of Varroa resistance monitoring is going to be in just grinding up DNA. So the good thing is for me is that you could just get some Varroa from your colonies, put them in a little vial and send it to us in the mail. Varroa are pretty durable when it comes to DNA preservation. So you could ship them in a tube in the mail, no refrigerant needed. So it's a way to expand our monitoring area. We're going to continue to expand monitoring. Like I said, this year I want to hit well, it is this year, it's 2023. I want to do 1,000 colonies. So if you know somebody who might be interested in running the test, I can get you the materials. We also need to evaluate new materials and methods for Varroa control. So finding out what is a consistently effective oxalic acid treatment, when to apply these kinds of things. What can we do with other materials that we have on hand? Because like I said, we need to start thinking about beekeeping in a post-Amitraz world. And then the really big important thing I want to start doing is distributing Varroa resistant stocks. So at the Honeybee Lab, throughout the last 25 years, we developed what's called Paul line. Paul line is a stock of bees that has Varroa sensitive hygienic behavior, which they keep Varroa levels very, when I say very low, I mean like less than 1 Varroa per 100 bees in a highly consistent manner. So those are another tool that we have to think about deploying in the fight against Varroa. So we can't just rely on miticides. We have to use multi-pronged approaches. So Varroa resistant stock and other cultural controls, I think, are going to have very big important parts in the ongoing war against Varroa. And so if you're interested in participating in 2023, you could email me and I'll ship you a free Amitraz resistance test kit. The only cost that you will have to incur is sending me the data. So you have my email here, and I dropped my email in the chat. And yeah, I can send this to you, no problem. And I also will send you the protocol. There's a video, data sheets, and I'm always here to answer questions. As you're going through and you're sampling any Varroa, put them in a tube or a vial. Give me the date, location, and contact information. I'll send you a shipping label. It costs us like $2 for you to put this in an envelope and ship it to me. So as you're doing your Varroa monitoring, it'd be great if you could send me those. If you have any old Varroa in the freezer, that might go back a couple years. It'd be really great to have, because what we want to find out is where the changes in the DNA, where these resistant alleles in the population that are just starting to get selected for, how have Varroa changed genetically over time? And unfortunately, I can't go back in time, so we might want to try to track some Varroa down in the freezer. And then I'm also doing the Apivar strip exchange program. So if you ever use Apivar and you feel like it doesn't work, send it to me and I'll run it against the Varroa that I know are susceptible. And that way we know, I could pretty much guarantee that those old strips or whatever strips you put in are going to work on the susceptible. So it's resistance. That's probably going to explain why they didn't die. And so I didn't do all this work myself. I'm really fortunate to have really great technicians, Victor there on the left, and then Dave serving up a whole helping full of Varroa. And Dave is my beast technician. We worked till like midnight some nights on these trips to get the research done. And then Nathan, see over here, they do these gigantic bioassays for us to get the data that we need for our projects. I worked with the Bee Informed Partnership, Anne-Marie, Natalie, and Cade. Spent 2 weeks on the road with them, and they help us to really get a lot of data across the country in a timely and cost-effective manner. And then Jeff Williams, I've been collaborating with on this project with his graduate students, Dan and Rogan. And so they will be doing some surveys on Amitraz use patterns to see if that leads to resistance. And then just all the people that have been helping out, cooperating beekeepers, apiary inspectors, other researchers, and then these organizations for funding and availability, raising awareness of this topic. And so with that, I'll take questions as we can for as long as you want. So
Yeah, Frank, I'll go through the ones in the chat and then you can just
Okay.
knock those off. And then if anyone has questions after that, they can just raise their hand or just start talking.
Yeah.
So someone said they thought CBPV was spread between adult bees through infected food and feces only.
no, so it can be transmitted. Yeah, that's true.
Right.
But it can
Maybe
also
not.
be transmitted by Varroa. Yeah.
Okay.
Yeah.
So Jana, I don't, Jana, was this question answered? I think it kind of was, but you said, what are the alternate
That-
miticides that could be used alongside of Apivar? And do we
That
use them
was not,
simultaneously?
that question was not from me.
Okay, sorry.
Yeah.
So
So
yeah, you
we are
got it.
looking at different combinations of of miticides. So Jeff Williams' student Dan actually just ran a test this year in which they used Hopguard and thymol together to see if there was any kind of, especially on these Amitraz resistant Varroa, just to see if that would work. And they got some interesting results, but it wasn't as really strong as a control. I think he's going to be writing that up pretty soon that I could talk about it more in detail.
Okay.
But yeah, like I said, we have some other tools in the toolbox. Like I said, oxalic acid, it's been used in Europe for 40 years. And if they can use it for that long with effective control, we need to figure out why it doesn't work as consistently here. I think a lot of it is weather, because in California, you can put oxalic acid and people swear up and down about it. However, in the Southeast, where it's really hot and humid, some reason it doesn't work as well down here. It's also another thing where those compounds are more effective when they have less brood. And down here in the South, we almost never go broodless. So we have to figure out the hows and whys to give more consistent treatments.
Okay, and then I think this one came from you, right, Brent? It says, do you feel like a club like ours should advocate for a similar treatment strategy among all members?
Yeah, I think it'd be really good, because I think what's driving this, and I'm pretty close to getting enough data to stick my flag and yell from the mountaintops that frequent application of Amitraz, I'm talking like 3 or 4 applications a year, leads to resistance. Typically in the past, people would just use 1 treatment, then 1 became 2. Well, if a little bit worked, more should be better. I think that's what's done it. So if you could organize those treatments on a way that uses Amitraz, but not in a way that's the only thing you're using, I think that'll really help slow down the development of resistance. So unfortunately, I really don't know what would be an effective miticide treatment up in Oregon yet. I'm sure Anthony would be really great to work with you to find that out, because Varroa control is so regional. But find out what works in Oregon, and if you could do it by rotating, and then also using non-miticide stuff. Like I said, these Varroa resistant stocks I think are really important for the future of beekeeping. And that's the other thing that we have to work on, is what are more consistent and low cost IPM methods. That way we could address the concern.
One quick question, a follow-up question on that.
Yeah.
If we were going to try to put together a strategy, what would be a good way to to begin that process? And would we interact with you, for example, to start that process?
So my academic grandfather is the one who came up with insecticide resistance management theory. So it's kind of in my DNA. So that's what we're trying to work on to adapt that for Varroa. So what I'm thinking is kind of, and this is all going to be in this review paper I'm working out, is to mitigate Amitraz resistance is rotations. So maybe 1 year use Amitraz, following year use oxalic treatments, following year use something else. So kind of like you do crop rotations, right? You use miticide rotations. And what that does is if they do develop resistance to one, that other thing, that the other miticide that you're going to use has a different mode of action, and it'll kill them out. So that way they're always guessing at what's going to be hitting them. So miticide rotations will be really good. That's probably a good thing. And then also too, doing it in a coordinated manner. So that way, all colonies are being treated. So if you think about it, if you're a large scale beekeeper, they're not treating 1 or 2 colonies in their apiary. They're treating all their colonies in the apiary. And while some people say, well, you're doing a prophylactic treatment or a treatment that might not be necessary. However, unless you're going to monitor every single colony in your operation, you have, I'm going to say if you're a large scale beekeeper, 10,000 colonies, you have to make sure that you have to assume they're all going to be treated. And that's really important, especially on a regional scale, because what could happen is that, especially if you could potentially have drifters from nearby colonies, but I think they have to be really close. I think the data I showed shows that geography really is important. Now, while that geography might not be important, I know that clubs like to share equipment a lot and share bees. So if someone loses bees, you might give them some bees. I think that the facilitated dispersal of Varroa might be important here too. So if you're sharing bees or equipment, make sure that you're knocking down your Varroa before you're sharing it, and maybe doing these tests to know that you're not distributing Amitraz resistant Varroa, because some people I've talked to have Amitraz resistant Varroa, but they never used Amitraz before. So they inherited resistant Varroa. So if you did it as a club to make sure that all the bees that you're sharing don't have resistance, that would probably be beneficial to all the members.
So how about seasonal rotation of treatment?
Yeah, seasonal rotations can work, and that's actually what's really recommended. Cameron Jack down at Florida did exactly that, where they used I think like 7 different miticides in the spring, summer, fall, and winter, just to see what's regional variable. So oxalic acid tends to be best. Oxalic and formic tend to perform the best when it's cooler out, because those materials can be hazardous at high temperatures, but they're also more effective at cooler temperatures because there's less brood for the Varroa to be hiding out in and escaping treatment. So yeah, so rotating. So in the winter, early spring, when you have little brood, it might be a good chance to use an organic acid such as oxalic acid or formic acid. Then when you get into the summertime, it gets a little tricky, because a lot of times in the summer, that's when you're producing honey, right? And no miticide is labeled to be used during honey flow. Oxalic acid is changing, but that's a state thing. I can't make any blanket statements about that. I don't know what it is like in Oregon. Check with your Department of Ag, or Anthony might know that. That would be something else. So yeah, that's regional and seasonal things would be good. Other thing you could do that's seasonal Varroa control is when you make your splits. So let's say you have a colony and you split it into 2. The old queen stays behind, and then the new colony that gets either the virgin or a queen cell, that's going to have a broodless period. During that broodless period is 2 things. 1, it's a good treatment opportunity for organic acids, and 2 is that's going to slow down the development of Varroa populations. Since Varroa tend to go slow, then late summer, August, it starts to get exponential growth. So if you could delay that in the springtime by keeping them broodless for like a couple weeks before they start to lay, that's another thing you do. So splitting bees is Varroa control as well.
So my understanding is that Formic Pro can be used during honey flow. Is that not true?
I really don't know about the label on Formic Pro. I'm not sure, so I'm not going to speak to that. But check the label. The label will have it on there. I'm sure it will. And I think did Hopgar get also a label change to be used during honey flow? I'm not sure. It's such a fluid situation, because that is a really important treatment time. And so we've also done some work looking at the application of oxalic acid during honey flow. And I think what's been interesting is that we're finding out is if you use it, if you get oxalic acid and mix it with glycerin, and it's all coming out in this paper from University of Florida, Cameron Jack, is that it won't knock your Varroa levels down in the summertime. It won't make them decline. It'll hold them low. So if you start with 1 Varroa per 100 bees, you'll end the treatment with 1 per 100 bees. So I think when we think about Varroa control, we don't have to think about like extermination or eradication. We need to think about holding populations in check. That is also Varroa control too.
Got a couple more related to the, let's see where we're at. How close are you to finalizing the Varroa resistant stock, and what are the biggest factors to distributing it?
So one of the biggest factors of distributing it is just a couple things. First is, and I'll be completely transparent about this, they don't make as much honey. We've done this a bunch of years, and when conditions are good, they'll make comparable levels of honey. So yeah, good clover flow and good rainfall. Let's say a comparable stock will make 100 pounds of honey. These resistant stocks might make 90 pounds. However, when it's a bad year, they might make 50% honey. So there's a trade-off there. However, that is a honey trade-off. The other trade-off, though, is that they tend to survive at a significantly higher level than unselected stock. So if the name of your game is to keep your bees alive, then that is probably something that you should consider in your economic calculus. You might make less honey, but your colony is more likely to survive. And you don't have to treat for Varroa as much. So there's another potential cost savings as well. Other thing that's really difficult is managing expectations. That's that's the name of my autobiography, is Managing Expectations by Frank Rinkevich. Is that, so this behavior is really complicated. So it involves these nurse bees detecting cells that are infested with Varroa mites, uncapping the cell, removing the pupa, and disposing outside the colony. So it's chemical, it's behavioral, it's all this kind of stuff. So it's controlled by a lot of different genes. And I think the last time they looked at it, it was about 12 different genes. What that means is that if you get one of these really good resistant bees of this resistant stock, she'll perform well, she'll lay lots of eggs that are performing this behavior. But the second that hits the next generation, so her daughter queen goes out, mates with a non-VSH drone, that F1, the one that is a VSH queen mated to a non-VSH drone, it'll be about 50% of VSH expression, just like you would expect from genetics. But if that one outcrosses again, it's gone. So it goes away really quick. So unless you're replenishing your genetic stock, doing directed breeding, instrumental insemination, this trait goes away quick. And so it's one of those things that has to be actively maintained. The good news, though, is two things. One, this past summer, I saw that happen at scale. I talked to this one beekeeper. I sampled 30 of his colonies. I found 1 Varroa in all 30 samples, which is insane. And so I asked him, what's your stock? How are you using it? He goes, well, I've been getting this material in the last couple of years. We've been focusing on the breeding program, and now they're to the level where he's happy that it's stable in his population. He doesn't have to buy breeder queens. They don't have to make sure they have the drone populations they have in their colonies are self-sustaining. And he said, yeah, this is something that took, I think he said 7 years to get to that level. And this is a large scale commercial beekeeper, 20,000 colonies. So it can happen at scale. The biggest thing is having a drone saturation area so that you can do open mating. And the way that that works for us is at the lab, we have this big giant piece of land on the Mississippi that we work with the landowner. And so we know that most of those drones our queens are going to encounter when they go do their mating flights are going to be VSH drones. However, not everybody has access to, 20,000 acres, so they can set their drone saturation areas. So that is a significant barrier. The other good news, though, is that the Hilo Bee project that was out in Hawaii is undergoing a transformation where basically they're trying to rear these queens at scale in Hawaii to supply the commercial beekeeping industry. There's been some changes in the program, but the ultimate idea is to make those available at scale. So it'll come online, but like I said, this is a very difficult to manage stock because it does require constant reassessment, constant evaluation, and highly controlled genetics. So that is a major barrier.
Someone did have a question about the expiration date on the strips.
Yep.
They're saying, are you saying that the expiration date on the strips is not important?
Nope. So let me clarify that though. If you have a pack of Apivar that's unsealed, it should be just as efficacious as ones that are brand new. Let me see if I can pull up that data real quick. I've tested strips that are manufactured in 2014 that work just as good as strips that were manufactured in 2021 in our resistance assay. I haven't applied these at the colony level, but like I said, if they're effective in our resistance test, they should be effective at the colony level. That's just been my experience. So what do I have? I have vintages of Apivar going back to 2014. I got some for 2017, and I have a lot of other people sending me in strips from, oh, this stuff doesn't work. When I get it in my colonies where I know I have stuff with Varroa, it works.
Interesting.
The one thing I just do want to indicate though is that if you get Apivar and you apply it to your colony, do not reuse those strips. Those strips, reusing them will not work. I need to be very clear about that.
Okay. One person asked if the queen could have been the factor in the differences you saw amongst different beekeepers.
So in terms of Varroa levels, I would say probably, because like I said, there was that one in that graph, there was the one beekeeper who had 0 Varroa, I think beekeeper C. So yeah, that's very important because you can't have Amitraz resistant Varroa if you don't have any Varroa. A lot of the people that we worked with in this study, they really didn't know too much about their genetics. So typically they're just breeding from their survivor stock within their own operation. Whatever that is, is it a little bit of Italian? Is it maybe a little bit of Russian in there and some other stuff? We don't really know. I really think that so that is about Varroa levels, but the levels of Amitraz resistance in that Varroa, it definitely comes down to how much they're using Amitraz.
Okay, we have a couple different questions. I'm trying to go through here and consolidate them. Do you feel like the lack of foraging plants for bees causes more Varroa mite infestation?
Debatable. There has been some evidence to suggest that, but I don't think it has been as strong as I would be comfortable with. So there has been some data on that. So access to good forage also prevents the development of, I think it's Israeli acute paralysis virus. I think that's what it is. But I can't really recall any study that showed that very significantly. There very well might be something out there that shows better forage leads to lower Varroa populations, but I can't think it off my head. That being said though, better forage I'm a big advocate for it because without good forage, you can't do really much of anything. So if bees on good forage have less Varroa, to me, that's like sprinkles on top.
We had a couple questions about the strain of Varroa resistant bee strain. I wonder if you could maybe put the strain name in the chat and if it is available at this point.
So it's called Pauline, and that stands for pollination line. That's all it stands for. And this was a project that started off 20-something years ago, and it has high levels of VSH behavior, but it also has high desirable characteristics of having large populations of almonds, higher winter survivorship, and like I said, in good years, comparable honey production. The other thing I really like about this stock is that they are so gentle to work with. You can work them in just a veil and a t-shirt. Fantastic, especially out here where it's super hot. Now, that being said, is that we can't make them available. We don't sell them either. However, there are people who can sell breeders. So for example, like Joe Latshaw has this material that a long time ago and developed his own stocks off of it, Adam Finkelstein, and things like that. So there are people out there with these genetics in their stock. However, because I feel like there is a little bit of a bottleneck, that is going to be something that I'm going to be working with next year, next 5 years, is to get some of these genetics out to clubs like yours. So that way you can get some of these genetics in your populations, and over time, hopefully they'll become self-sustaining. So I think the distribution of that material is going to be changing in the next 5 years.
And while we're talking about this, someone just asked a question about are they better pollinators? I mean, you talked about the honey production and their gentleness, but what about pollination?
Oh, you must have followed me out to California because that's exactly the question we were asking this year. So we just got back from California on Friday. We spent 2 weeks out there working with this group called Bee Hero. They have these little sensors that they can put on the colonies and tell you how many bees there are and how many are foraging in and out. And so we're comparing Paul line to Russian bees and then to a third unselected stock. And so what we wanted to find out is, do any of these stocks forage at earlier times in the day, at cooler temperatures? And if so, which ones bring in more pollen? So on a colony by colony basis, standardized by size, which ones are more efficient foragers? So we just had that data in on Friday, and unfortunately I haven't analyzed it in a couple of days, but that's something that we're going to be publishing. So preliminarily though, I don't really see them as being better or worse. They seem to all be equal, but that's just this first glance at a billion data points, because those readers take data every minute and then continuous sampling through the day. So you get this huge set of data. So it's a lot of data to go over. And so we're gonna have to focus on that. But yeah, we're gonna find out if they are better or worse. We'll just see what the data says. It's coming up.
Someone asked another great question. I didn't think about this. So since Amitraz stays on for 40 plus days, based on your test, the majority of the effect is in the first day?
Yeah, so this is a little bit different. So in my example, these bees are in a confined space where they have no other choice but to interact with that Amitraz strip, right? But then when you think about when that strip is in a colony,
that makes sense. Yeah.
2 strips for brood chamber, they don't have the probability of them interacting with that strip is significantly lower. So it does take a little while for them to start to fall at the colony level.
It's the confined space that's really forcing it on them in that first 3 hours you do, right?
Yeah, 100%. So I mean, if you think about it, the equivalent would be like replacing all your frames in your colony with Amitraz strips. So yeah, so the probability of them encountering Amitraz strip in a colony is significantly lower. That's why it takes a little bit longer time.
There's a lot of questions in the chat here, and I may have skipped around a bit. If anyone has another question, feel free to just chime in and ask your question now.
Yeah.
Oh, the one at the bottom is a good one here. Where can we get the testing kits so we can make our own, or can we make our own testing kits? It looked like your test was really simple. It seems like something that we could do on our own.
Simple, cheap, disposable, reproducible. If you could have a bioassay that's those 4 things, you got a winner. So yeah, I could send you I could drop it. I could send it to you to distribute to the group. But this test cost, let me just pull up the data so I can have it here. So I'm speaking accurately. Give me 1 second. Okay.
And while you're pulling that up, I mean, data points like this on an individual apiary is not as useful as it is in the larger scale, right?
Well, believe it or not. So then the other thing is, I didn't mention, is that I think so let me just say this statement, is that within an operation, resistance across apiaries within an operation tends to be similar. So if we go out to a beekeeper, sample 1 apiary, get a data point. We go to the next one, it's very similar. A third, similar. So it's pretty consistent across a beekeeping operation in which the Varroa are managed in a similar way. So that's why I usually say 2 apiaries at most, because they're very similar within an operation.
Just a quick question. Do you do any YouTube videos on any of your research results?
I actually have a YouTube video about the testing protocol that you could look at, and I'll send you an email with the link to the video, the protocol, and the data sheets, so you can see what's getting involved. And then you could also see what's in these resistance testing kits. So let me just pull it up here real quick.
Yeah, anything you want to send to me, I can ship it out to this entire group.
Yeah, that'd be the easiest thing. I could get that out real quick. Yeah, so let me just give me 1 second. I'm still working on getting that thing pulled up. It's 2020. Equipment costs. Here we go. So equipment costs for this study is, when you look at all that stuff, it costs. So when you do your first apiary test, let's say you're going to test like as a group, you're going to test them. It costs $27 in parts to sample 10 colonies. That's extremely cheap. So that's 10 colonies per apiary. Now, let's say you want to do some more in your apiary, other apiaries. There's a lot of equipment that can be reused. So the cost per apiary, based on what you can reuse in this test, is $12. So it's stupid cheap. And like I said, I get a lot of this stuff from just restaurant supply places, home Office Max. It's not fancy. It's very DIY, and that's what I love about beekeeping and honeybee science, is that it's a lot of just, okay, let's make it work and improvise something. So yeah, so I have all those parts. I got the prices. I got everything that you'll need to do the test. And so if you wanted to do this as like an organization, I mean, it probably wouldn't cost too much if you want to do that. Or I just send you the equipment and you can do the test that way. So either way, we could get some data from it for sure.
Great. I got a few more questions here. It says, could you say a little bit about the effects of underdosing and overdosing Amitraz?
Yeah, good question, because those are two different aspects on the selection regime. So you underdose, right? It's so selecting for resistance, the Amitraz resistance of Varroa is almost exactly like selecting for antibiotic resistance in any kind of pathogen. You underdose it, it allows a higher proportion of those target individuals to survive. So instead of wiping out the population with a high concentration, you're going to hit a lot of them. Most of them are going to die, right? However, you're going to let a lot of them slide through. And that's typically a little bit more problematic when you develop resistance, because what happens is when you have a lot of survivors, those survivors could have all different resistance mechanisms. So that way you could select for multiple resistance mechanisms. So when you underdose and you get resistance, it's typically much more problematic. Now at the other end, and so when you're when you're underdosing, the resistance will typically develop very slowly and gradually over time, where just over time it just keeps increasing that way. Now, when you're using high concentration, that's typically referred to as management by saturation, where you give them such a huge dose that there's the probability of any individual surviving is very, 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 very low. However, you do find that one individual that has that beneficial mutation, and then it just takes over. The problem, the benefit to that is typically you're only select for one resistance mechanism. The downside to that is that it usually makes them invulnerable. And what's going to happen to resistance is over time, with that high concentration, you're going to see no resistance, no resistance, no resistance, no resistance, then all of a sudden a little bit, and then it's like up here really quick. Maybe that's what's happening now, because what we're starting to see is we're starting to see more prevalence and much higher levels. So I don't know if that's where we're at. And the really unfortunate thing about this with Amitraz is that you have to make sure that you're using 2 strips per box. So that way you're getting the proper, I guess, dose for the bees. Don't cut the strips in half. Don't take them out before that 42-day prescription. Treating as according to label is really important for managing resistance, because that information is not just arbitrary. It's made up of, how long are they going to kill Varroa for, and what is the dose that's really going to be very high level. So it's definitely worth your time to read the instructions and apply it to the labeled instructions.
I got one more question for you. It says pesticides were not a big factor in the graph that you showed. Is this due to better pesticide practices? Were the pesticides still causing colony loss? And what are the major factors contributing?
Yeah, we didn't look at pesticides in any of these studies. Yeah, so I really don't know about the effects of pesticides on our study system. In a totally separate study, we did look at the effects of pesticides And the way that we did the sampling was we standardized the pesticide that we tested based on controlling for Varroa levels. So that way we knew that Varroa wouldn't be an issue. And when we did that, yeah, pesticides were a problem, and it maybe affected, I think the whole number was 17% of the colony losses could be potentially ascribed to pesticides, but we could ascribe 70% of colony losses to Varroa. So yes, pesticides are a thing, but the effect of Varroa is significantly higher.
Cool. Sorry if I missed any comments. If anyone wants to chime in with their question that I missed, feel free. If there's any other questions, that's all I have for right now.
Can I ask an unrelated question?
Please.
I'm curious about these hives that I've been staring at during the end of the presentation. Where are these hives, and is this normal to have that many supers stacked on hives?
Yeah, you can make lots of honey in the Dakotas. Yeah, that's typical. 6-7 boxes high, 200 pounds. This was a good year. We had some reports of 200 pounds or more per colony in some areas. It wasn't everywhere, but if there was a good amount of rain, not too much, not too little, not too much kind of thing, yeah, this is what you'll see.
Wow.
Yeah.
Thank you.
Well, if there aren't any more questions, that was absolutely fascinating and informative. Thank you so much. There's so much to talk about and so much to learn, and we're excited about your research, and we hope to stay in touch and watch how you progress through this, and perhaps become a partner if we can figure out how to do that in an effective way. We'd like to, perhaps send you some information and help your cause for sure.
Good. Yeah, I think this is something that resonates with a lot of people because we all know that Varroa is a big, important problem. So yeah, any kind of questions that anybody might have, send them my way, and I'd be more than happy to talk with everybody. So thank you very much for inviting me and attending the meeting. It's always good to hear lots of questions and engagement. So yeah, it was my pleasure.
Okay, thank you.
I just want to interject here. We're from Tillamook, and I just want to thank the Portland Urban Beekeepers for including us in this. Thank you.
Thank you.
Thank you.
Okay. Can you all see our screen now?
Yes.
Okay. Wow, that was fascinating. So much to think about, so much to talk about. Okay, so let's move on. We have oh, I'm sorry. We're going to have a Yellow Jacket management talk with Brian Fackler. Brian, are you ready to start?
I think so.
Okay.
Do I?
Stop sharing and share with you.
Let's see. See, is that? Let's see, view.
Can everybody see Brian's slides?
Yes.
Okay.
Okay, well, I thought today was this time of the year is a really good time to talk about yellow jackets, because very soon the queens will be emerging, and if you can control the queens, you've got it pretty much beat for the year. So tonight we're going to be talking about yellow jackets, and our agenda are: what are yellow jackets? How to identify them? What are their foraging practices? Their life cycle and behavior. We're going to talk about integrated pest management for yellow jackets and the importance of cultural controls, physical controls, and chemical controls. So the identification of yellow jackets, excuse me, I got to flip my page here. They're a common name for a predatory wasp. and they're distinctive with alternating black and yellow stripes. The workers are approximately 1/2 inch long. The queen is approximately 3/4 of an inch long. They have a narrow body with a pinched waist, a hairless body, and smooth legs. They are fierce and the most common insects to sting humans. The stinger barbs are smaller than the honeybee, and they can usually be withdrawn and sting again. And I gave some examples of the comparative sizes between yellow jackets, honeybees, paper wasps, and you can see those. And I'm sure you've seen them in your apiaries or your yards. So yellow jackets forage for sugar in the form of nectar, fruit juice, and tree sap. They seek meat protein to feed their young. They chew up meat, insects, and fish, and in return, the larvae exclude a sugar substance the adults consume till about the end of August, when there's a few larvae in the nest. And at that time, the adults switch from meat brood to brood sugar for their own food. So the life cycle and behavior, yellow jackets are semi-social colony nesting insects. In the spring, overwintered queens emerge and select a new nest site, an underground hole, an opening in a structure, or an aerial nest site. They build a gray paper nest from wood and vegetable fibers that are collected and chewed by worker yellow jackets. The queen stays in the nest and lays eggs the entire growing season. In August to October, new males and queens are produced and mate. The mated queens will find a location for overwinter dormancy. After the temperature drops to around 32 degrees Fahrenheit, the colony dwindles and dies. In late summer, the yellow jacket population is large, and the typical food supply is declining. And that's when we experience the aggressive savaging behavior. Oops. And they become a nuisance to both honeybee colonies, where they rob honey and larvae. So for integrated pest management is a system that includes cultural controls to ensure strong colonies and a good technique for preventing and controlling yellow jacket damage. The cultural controls that you need to do is to maintain a strong colony of honeybees, which will assist in keeping the robbing yellow jackets from the beehive. This includes frequent inspection by you, the beekeeper, and actions to control Varroa mites and maintain a productive queen are essential to a strong colony. Physical controls. In late summer, you can reduce the entrance and or add a robbing screen. That'll help protect a small colony from robbing. Also close the upper entrance from March and from March to late October, install multiple traps. And I've included the one trap that's probably the most common around is the rescue yellow jacket traps. You can buy these in either disposable form, or you can buy one that's a reusable form. And if you're using the reusable one, you want to replace the trap and the bait approximately once every 10 weeks. And I've used these for years, and they're very effective and not very expensive. For chemical control, from early March to late October, go to your grocery store and purchase the greasiest deli. I use drumsticks. Add a small amount of both liquid yellow jacket attractant and Onslaught insecticide. And Onslaught is the only licensed insecticide for baiting yellow jackets, and this is recommended by Oregon State. You place these in a bird suet feeder, which are hung near the hives. Birds are not meat eaters, and they won't you don't have to worry about your bird population. Each month, I replace the treated chicken the entire season. The yellow jackets chew the treated meat, then return back to the hive to feed the larvae, and the poison on the meat effectively will deter or kill the whole colony. So key points to take home. Yellow jackets are both a threat to humans and honeybees. You can control yellow jackets with integrated pest control. Success requires your proactive season-long effort. Your bees will appreciate your control of this pest. These are my references. I went to honeybee biology and beekeeping, which we're all familiar with. Excuse me. And the Canadian Honeybee Diseases and Pests is a publication that comes out of Canada. It's probably one book that you need to buy for your library. Another really interesting book is by Clarence Collison out of, he was a former professor at Mississippi State, and he wrote a series of articles in bee culture for many, many years, and this is a collection of his just tremendous good information. The newest edition of ABC and XYZ of bee culture, I use that. And then I use two publications out of Oregon State as my references. And so I'm open for any questions.
Yeah, Brian, I got one question for you.
Certainly.
On this property that I'm on, it has like a 3 or 4 year old fig tree, and I was told that's a really big attractor to yellow jackets. Do you know if that's
I don't know about that.
okay?
I know that if you get yellow jackets in your area, they will be nesting in the area, and you're going to have a huge problem until you control them. And I had a huge problem at my house in Vancouver. We had timbers that were on the side of my property that were like railroad timbers, and we had yellow jackets nesting in them, and I had to pretty much eliminate those timbers to get rid of them, because I tried many years in a row to get rid of them and it didn't work.
Yeah, thanks.
You're welcome.
Where do you get the yellow jacket attractant?
I get mine at Home Depot.
Okay.
You can get it from any hardware or garden supplies place. They'll have vials of yellow jacket attractant. I know Ace Hardware has it in Lincoln City.
Thank you.
You're welcome.
Brian, when you did you see yellow jackets when you did inspections, or how did you know that the yellow jackets were robbing your hives?
Okay, when I first started having bees at my beach house in Lincoln City, there was quite a bit of yellow jacket activity, and you could see that in the late August, you just saw a tremendous amount of yellow jackets. I think the first year I was a beekeeper, I had a tremendous amount of yellow jackets around my hives. And, we commonly eat outside on picnic tables, and you'd open a can of pop or have some, and they'd be in your food and in your drinks and forcing you in the house. And I pretty much knew I had a problem. And years ago, I remember going to the Tillamook Bee Club, and there was an old time beekeeper that kind of clued me into this method of doing it. And then I realized that very soon that, hey, these are the recommended products, and this is what Oregon State recommended, and on and on. And so I've been using this for approximately 10 years, and I virtually have no yellow jackets now, and I've kept them under control. And I haven't had to use the bait method in my house in Vancouver, but I certainly catch them in traps.
Okay, thank you.
You're welcome.
So there was a question in the chat about, is there no good purpose for yellow jackets? And
You know, all the other wasps are beneficial pollinators and shouldn't be killed. So unless they're a threat to you, but I can see no beneficial benefit for yellow jackets. I think they're too much of a threat to humans and to your bees. They are fierce, aggressive insects, and no one likes to get run out of their yard because of the threat of getting stung or attacked by a whole swarm of yellow jackets. And you shoo one away and they'll come with a vengeance, a bunch of them. So I can't see any beneficial reason not to kill them, for them to be around. That's what I meant.
Okay, excellent. Well, Brian, thank you so much.
You're welcome.
That's really helpful. We've been so engaged with all of our speakers today that we're probably running a little bit low on time, but Jana has some really important information to share with us. So I want to make sure to get to that. And Jana, let's see here.
I think
Brian, if you can just stop sharing your screen, I think I can get that to Jana.
let's see. I don't know how to do that.
Oh, okay. Well, can anybody help Brian with that? Maybe I can do it from my end.
Brian, where you click to share your screen to start with,
Yes.
click on that again, and then like down at the bottom of the screen, there's a share screen button.
Yeah.
Click on that, you should have an option to stop sharing.
Did that do it? Oh, I see it now. I found it. There you go. Sorry about that.
Okay, Jana, are you ready to go?
Yeah, can you start sharing and pull up the hub slides for me?
Oh, yeah. I thought that I put you on there. Okay.
Oh, there you go, perfect. Okay. So the swarm list is still, we're still adding people to the swarm list. So if you aren't on it and want to get on it, you can see the registration code there on the slide. You only need that code if you have never registered before. Or the other possibility that you would potentially need it was if you were on it last year, but your dues lapsed and you didn't renew, and you got kicked off the swarm list, but you subsequently renewed your pub membership dues, and then want to get back on the swarm list, you'll need that code. We've got really cool filters now on the swarm list, so you can really dial into what kind of swarm situations you are willing to travel to. So it should cut down and make your swarm calls a lot more efficient this year. Oh, thank you, Victor, for attending from Cowlitz. We really appreciate you guys coming over.
Thank you so much, Victor.
We are starting a brand new swarm mentorship this year, and I'm super excited about it because I personally learned how to catch swarms for the first time last year and had a blast. I'm so addicted to it, and I am all about helping everybody else in the world learn how to catch a swarm too. So if you are somebody who would love to learn how to catch a swarm, we emailed out a couple weeks ago a registration form for you to, oh, great, Daniel, we're glad you were here from Portland Metro too, thanks. We would love for you to fill out that registration form. If you did not get one and you want one, hit me up at swarmlist@portlandurbanbeekeepers.org and I'll send it to you. But in order to help our students, we need more mentors. So currently we have 4 mentors, including myself, who are willing to have new beekeepers come along or new swarm catchers come along to learn how to do it. But we have like 7 or 8 students who would like to learn. So I would love it if there's some experienced swarm catchers out there that would be willing to let students tag along and watch how the process goes. Keep in mind the mentor gets to keep the bees. So mentors, potential mentors, it doesn't hurt for you to have someone come along to lend a helping hand to catch the swarm and or just watch and see how it happens. If you want a mentor, again, just shoot me an email at swarmlist@portlandurbanbeekeepers.org and I will get you set up with that form. Okay, so a couple of things on the apiary. Originally, our plan was to start the first Sunday of March. Our normal days are the first and third Sundays of the month out at Green Acres to go to the learning apiary. It's going to be way too cold this coming weekend to open a hive. But what I would like to do is invite people to give me some feedback here and now, either in the chat or just verbally pop in. I am willing to go out to the apiary, give people a tour, do sort of an orientation, and then do some things like I can show people all the different types of hive equipment that you might want to have. You know, some of the more unusual things, how they work, why you might want them, why you might not want them, how to put it all together. You know, we can talk about different kinds of tools and equipment. We can talk about the kinds of different mite treatments you might want to have on hand for the year so that you just have it and you're ready to roll. You know, if you find you have a mite problem, you don't want to get, you know, have to spend time waiting to get your treatments. Just answer any questions people have about getting new bees or getting the year started right or anything like that. I'm totally happy to go do that. I just don't want people to come out and be disappointed I'm not going to open a hive in 40-degree weather. So anybody have input on whether they would like that kind of an apiary visit?
I was really excited about that.
Okay.
I planned on coming. So
Okay. Anybody else interested in that kind of a day?
I would be interested as well. I would definitely come.
Okay, 2's enough. We'll do it. 11 to 1 at Green Acres. And if we go longer than 1, we go longer than 1. And if we're done at 12:30, we're done at 12:30. So come on out, and I think it'll be really fun.
I think so too.
I am also this year going to be starting an email list for people who are interested in the apiary, so that I can keep that core group of people really super informed about what's going on. If there's changes to the schedule or anything, I mean, I'll try to keep the club as a whole informed, but I also want to make sure that the people who are like the core people who are continually coming and dedicated to coming really have, you know, that sort of extra hands-on thing. So if you are interested in being on an apiary update email list, please shoot me an email at apiary@portlandurbanbeekeepers.org. And I'm getting that list put together now. Does anybody have questions about Swarm List, Swarm Mentorship, or coming to the apiary?
I'm sure I'll have lots of questions when I see you out there at 11 o'clock.
Cool. I hope I'm ready for them.
Yeah. Y'all, we're going to have a little bit of a time for general questions and answers right now. And of course, that can include questions you have for Jana. I do want to acknowledge everybody staying engaged and staying with us tonight. It's been a wonderful members meeting, I think. Are there any questions for us tonight? Okay, well, then,
I'll ask another stupid question about those beehives. So is that normal to have that many supers on a hive?
Not
As was
here,
in the,
not
huh?
here, Christy, not
Okay.
necessarily here. I think that was probably a commercial beekeeper. Or like a sideline beekeeper who's more than just hobbyist. And I think he said it was in the Dakotas.
Right.
So I think like people maybe in the South too, where they have a really long, warm growing season. But around here, most people don't end up with 7 or 8 supers.
Yeah, there were 9 on one of those I was counting. It was like amazing. So
Yeah. Yeah. I mean, that's for me, that's wishful thinking. But I suppose it probably kind of depends on our weather too. I mean, if and where you live, like, if you live near like major farmland and orchards and stuff that have varying blooming cycles, and they're large operations, and and if we had a super warm year, maybe you could get several supers, but we don't usually see 9 supers
okay,
around the
and
Portland area.
so another question then, at what point do the bees have to go so far up that they would prefer just to swarm or something? Do they just keep moving into those supers, or do they say this is just too much work and I'm out of here, and then swarm out?
Well, the swarming is typically triggered by lack of space.
Right.
So, I suppose if they got honey bound, if the queen got honey bound and she had nowhere else to lay eggs, maybe they would. Is Dewey still on here? Dewey, are you still with us?
No, I just checked and he's not.
Ah, shoot.
He can
Glenn's on
answer that.
here. Glenn's got a ton of experience. Maybe Glenn can answer that. Are you still here, Glenn? Susie? Brian? One of you guys?
What's that?
Do one of you guys have an answer to that question? Christy was asking if the bees ever decide that there's so many honey supers on and they have so much honey that they're just done and they're going to swarm out.
Well, no. The quick answer is no, that swarming is going to happen not during a honey flow, probably. Swarming happens for different reasons than that. Read Dewey's book. He's got great stuff about that. He'll answer you all your questions.
Okay.
I have one quick question. This goes back to Frank's presentation. Did I hear, or what did I hear that when he was talking about beekeepers or apiaries that were side by side that were affecting mite resistance in the area? I'm going to have to go back and watch the video, but did I hear something like that?
Geography was not a factor.
It was
meaning
not a
that
factor.
meaning that just because you had high concentrations of hives is what I took from it, is that that did not increase the Amitraz resistance.
Okay, good. Thank you.
Right, that the geography wasn't a factor, but when one beekeeper had a number of apiaries within, a close distance of each other, that then there was a correlation between those because it was run by the same beekeeper. I think that's what he was saying.
Yeah, and it was also the same practice versus a different practice, right?
Yeah.
Right, it came down to the practice. Yep.
Interesting. All right. That was a great presentation, both Frank and Brian. Y'all, thank you for tonight. Are there any other questions?
Nancy, check the chat. I gave you some membership information. And Glenn, I am so impressed that you had you got 10 supers of honey off of a hive. That is unbelievable. I'm assuming that's like way more the exception than the rule though, huh? Oh,
I don't know. I don't know if his I don't know if his mic is working, but that is a lot of honey.
that's
366 pounds.
that's unbelievable.
Yeah.
Love it.
Oh,
All
and
right, then.
while one thing, while Glenn's on, Glenn, if you can hear us, I think that we would like to do the pollen and nectar report next month if you're available.
You know,
You can just shoot me an email or drop in the chat.
I don't see him on the list. I wonder if he boogied out. Oh no, he's still on the list.
Yeah. Just maybe his mic isn't working.
Yeah.
But I'll send him an email if he didn't hear that.
Yeah.
Okay. Thanks, everyone.
All right, everybody. Thank you so much. We'll see you again either at the apiary or next month.
Thanks.
All right. Bye-bye.
Bye, everyone.